Hi, my name is Chyntia Cabezas, and today I will be talking about how the flu vaccine is developed and its effect on our body. I would like to begin this presentation by talking about some influenza milestones. In the 1900s, the flu virus H1N1 hit and caused a global pandemic. In 1930, scientists discovered that the flu is caused by a virus and not a bacterium. In 1957, the H2N2 virus emerges and starts a pandemic replacing the H1N1 virus. In the 1960s, the U.S. Public Health Service recommends flu vaccination for people who are high risk, and a new H3N2 virus emerges replacing the H2N2 virus. In the 2000s, the genome for the 1918 flu virus is fully sequenced, and a new virus emerges to trigger yet another pandemic. So, how are the flu vaccines developed? Well, there are three types of flu vaccines production technologies approved by the U.S. Food and Drug Administration, otherwise known as the FDA, that are developed for commercial use. The first one is the egg-based flu vaccine. This is the most commonly used manufacturing process to make a flu vaccine. This process makes both inactivated or killed vaccine, known as the flu shot, and the attenuated or weakened vaccine, known as the nasal spray flu vaccine. This egg-based production process starts with the CDC or another laboratory in the WHO Global Influenza Surveillance and Response System, providing private sector manufacturers with candidate vaccine viruses, or CVVs, grown in eggs per current FDA regulatory requirements. The CVVs are then injected into fertilized hen's eggs and incubated for a few days to allow the viruses to replicate. The flu-containing virus is harvested from eggs. For the flu shots, the vaccine viruses are then inactivated and the virus antigen is purified. Then the process continues with quality testing, filling, and distribution. For the nasal spray flu vaccine, the starting CVVs are live, but weakened viruses that go through a different production process. The FDA tests these vaccines to approve them before releasing them and shipping them for use. There are some downsides to this method because the production using the egg-based flu vaccine requires a large amount of chicken eggs to produce the vaccine, and it may take longer than the other methods to produce. The second method is the cell-based flu vaccine. This method is used to make inactivated flu vaccines, known as flu shots. To make this vaccine, the CDC or another laboratory partner use influenza viruses that have been grown in cells to make CVVs, which are then sent to a vaccine manufacturer. Next, the vaccine manufacturer inoculates the CVVs into cultured mammalian cells and allows the CVVs to replicate for a few days. Then, the virus-containing fluid is collected from the cells and the virus antigen is purified. The manufacturing process continues with purification and testing. Finally, the FDA tests these vaccines to approve them before releasing them and shipping them for use. This method is advantageous because it does not require chicken eggs and it has the potential for a faster startup of the flu vaccine manufacturing process. The third method is the recombinant flu vaccine. This method does not require a CVV sample to produce. Instead, recombinant vaccines are created synthetically. To make this vaccine, flu scientists first obtained DNA for making a surface protein called hemagglutinin, or HA, that is found on influenza viruses. HA is an antigen that triggers the human immune system to create antibodies that specifically target the virus. This DNA is then combined with a baculovirus, which is a virus that infects vertebrates, resulting in the recombinant virus. The baculovirus helps transport the DNA instructions for making flu virus HA antigen into a host cell. Once the recombinant virus enters an FDA-approved host cell line, it instructs the cell to rapidly produce the HA antigen. This antigen is then grown in bulk, collected, purified, and then packaged as recombinant flu vaccine. Finally, the FDA tests these vaccines to approve them before releasing them and shipping them for use. This is the fastest method because it is not limited by the selection of vaccine viruses that are adapted for growth in eggs or the development of cell-based vaccine viruses. Finally, how does the flu vaccine protect the human body against the flu virus? Well, The flu vaccine protects the human body by stimulating the body's immune system to make antibodies to attack the virus. There are some side effects to the flu vaccine, such as slight raised temperature, muscle aches, and a sore arm where the needle went in. Every year, a new vaccine must be developed due to the virus rapidly changing. This is due to antigen drift, which is a mutation in the virus that causes a new strain to arise. Here are some references. Thank you for listening to my presentation.